Bill, the eukaryotic cell, is having trouble with his eyesight. He can barely see 5 micrometers in front of him. This is getting to be a problem because glasses that fit eukaryotic cells are hard to come by, and his vision keeps getting worse. He did some research and thinks his problem is coming from a mutation in his gene A. He thinks maybe if he can reduce or eliminate the expression of gene A, his vision will go back to normal. The way he sees it, he has 2 options. He could try to eliminate expression of gene A via RNA interference or via CRISPR. But which should he do? RNA interference uses double-stranded RNA to shut off gene expression. The double-stranded RNAs bind to dicer, an enzyme that cuts the RNA into short segments, forming microRNAs and/or small interfering RNAs. miRNAs and siRNAs bind to proteins to form an RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The siRNA often has perfect complementarity to its target site, so it is very precise. Once bound, the messenger RNA is cleaved and degraded. miRNAs also direct RISC to messenger RNAs, but only parts of it pair with targeted messenger RNA. This means miRNA can target hundreds of messenger RNAs because it is less specific, leading to translation being inhibited or messenger RNA being degraded. That seems like a good option. What about CRISPR? CRISPR is a technique that originally came from a natural system used by bacteria to protect themselves from invading DNA, such as viruses, but has been developed to target other genomes and allow for genome editing. If he were to use CRISPR, he would produce guide RNA matching the DNA he wants to alter. The guide RNA forms a complex with a protein called Cas9. When the guide RNA finds its target, gene A, Cas9 cuts the target DNA. The DNA will try to repair itself, but this is very error-prone and the gene can be disabled. He could also insert a piece of DNA carrying the desired sequence for gene A, where the DNA was cut, instead of letting it mutate randomly. After lots of contemplation, Bill decides he will go with the CRISPR technique because he thinks it will be able to more precisely target gene A. If he used RNAi, it could be less precise because miRNA has variability in what messenger RNA it targets, and with CRISPR he can choose himself exactly how he wants gene A to be altered, by creating a specific guide RNA and inserting DNA of his choosing at the location gene A was cut. Here he goes. And just like that, Bill can see.